Boom! What's up, everyone? Welcome to Simulation. I'm your host, Alan Sakian. Now we're sitting down with Chen from PrimeVax.
Hey, Alan, how you doing?
Thank you for coming on the show.
Yeah, thank you for having me.
Appreciate
it's a pleasure.
it. Yeah.
I love what you're doing with the public intellectual, trying to get more people excited about changing the world.
Thank you, my man.
Yeah.
Thank you. And we got you on the show talking about cancer cures.
Yeah. Definitely an exciting time for us, and definitely an exciting time for the industry. Very, very happy to kind of give like an overview of what the entire cancer treatment space looks like, where we fit into it, as well as what some of the other players in the space are doing, for which there are many.
Yeah, biotech's exploding right now.
Oh my gosh.
And everybody it's very relatable to people when it comes to people's health, because they want to know how they can live longer, healthier, all that kind of jazz. So that's probably the most applicable. But so wait a second, before we get into that, because I think it's really important, but why do you care about biotech, and how'd you pick this cancer? How'd you pick that?
Yeah, you know, it's funny actually. So I fell into the pharma and biotech world right out of college. Studied econ in undergrad. Didn't really do any real science. But then, you know, all my clients, when I was a strategy and a management consultant, they were drug companies. Over the years, I learned the industry just by the simple fact that I was working in the space for this time. And then a bunch of my friends started developing cancer. Not a good look. I wanted to do something about it. And at the time, you know, rewind the clock maybe like 5 years ago, I was doing strategy consulting work for drug companies. You know, like the Bristol Myers Squibbs of the world, the Mercks, you know, the Pfizers, like those general, you know, pharma companies. Helping them launch drugs, setting prices, looking at how do you actually get the drug correctly placed onto insurance formularies, for example. And, you know, I just thought that the industry wasn't really like, it was making some amazing advancements, but if you were to look at some of the objective data coming out of those clinical trials or some of those data sets, they weren't like too amazing. You know, you might be looking at 1 out of 5 patients cured effectively in melanoma, for example, but those are still relatively low numbers. Yeah. So one of these smaller biotechs with whom we were talking, you know,
how can we get it to 5 out of 5 cured?
You know what, man? That's where we're heading. I think that that's exactly where the industry needs to go. We are doing our best to, you know, play a role in that process. Really excited about it. But the biotech science team came to me 5 years ago, and they had this very, very novel new idea for cancer treatment, completely the exact opposite treatment modality as every other drug company or drug, you know, development process. And I said, wow, like looking at this from a strategy perspective, looking at what drugs are actually, you know, going through the process of being launched and being paid for, being reimbursed, covered, you know, for insurance, you know, like you'll go to a hospital now and you can just get these new drugs that are already, you know, available commercially. Looking at those drugs and seeing, you know, where their limitations are scientifically, and looking at where these guys could be actually really changing the game, I said I had to get involved.
What does that look like for once you pass mouse trials? Then you go to clinical trials with humans, and then once you go through clinical trials, then you have to have a certain threshold. You have to meet that criteria. And then you go into actual
sales and distribution.
And when you do sales and distribution, you have to figure out how to work with insurance. You have to figure out all of that stuff before you can actually get a doctor that is an immuno-oncologist to prescribe you with a cancer treatment. And so then most of these drugs cost like millions and millions of dollars to go through that process.
The R&D.
The R&D
is very expensive,
And then so usually there's an outside company that's funding that process, because it's hard for average Joe to be to
build a drug, to build a cancer drug. Absolutely. Yeah. So I can talk to that a little bit. You're absolutely right. You begin first with mouse data. Once you generate that, and once you think that that looks good, you submit it to FDA, and then you begin the clinical trial process. Typically follows 3 phases: phase 1, phase 2, and then phase 3. Every step of the process, you're generating more and more data. It's more statistically significant. You can run analyses on patient subpopulations or mechanisms of action or what have you. And as you mentioned, toward the end of like the phase 3 timeframe is really where the rubber meets the road and you need to demonstrate real, actual efficacy, meaning statistically significant, directly, head-to-head, ideally, with a comparator arm with whatever the current standard of care is to show that, cancer patients are currently being treated at this amount with this level of response, and now we, with our new drug, are coming in with this much better of a response rate, for example. And then you submit that to the FDA. FDA looks at that and says, yep, this looks good, or no, this doesn't. Come back with more data. Ideally, you do that process, you get that done, and then after that, then you turn into the full commercial stage. Now, it's interesting that you mentioned, like the commercialization aspect of it and needing a larger external company to help do the sales and distribution, right? The funny thing is that, for us, where we came from, or where I used to come from as a strategy consultant, is that we would have to do this a lot earlier in the process than you'd think. Your commercial strategy for launching the drug kind of already needs to be built out and formulated during your phase 1 human trial. And ideally, you should be kind of building that into your earlier mouse models and other petri dish in vitro type experiments when you already have in mind what the future of the actual commercial applicability is going to be.
But it's hard to invest money into that if you haven't gotten the FDA approval that you can even go. So some people are worried that they would invest money into that and then not even get approved. Which is understandable, but at the same time, then if you're building out that model, it gives you a better understanding of what your drug's going to look like as it impacts the actual world outside of these mouse trials and clinical trials.
It's funny. I think that many people in the healthcare space are very micro-focused or laser-focused on that specific segment of their development. Some people in drug companies only do early clinical trials. Some people are only commercial people. Some people are only regulatory affairs, and they do nothing else but that. But I think for me, at least, where I come from, and my entire worldview on this drug development process is that you kind of need to know where you're going when you're much earlier, which we are. But thankfully, we're about to enter human phase 1 trials right around the corner. So it's an exciting time.
Exactly. So now the body is so complex. There's so many different ways for the body to get disease and then also combat disease. So there's some very simple way of understanding this with maybe you're getting a flu. You get the flu, and then within a couple days, your immune system fights off the flu.
That's right.
And then you're good to go. But there are other times when your cells become cancerous cells, and that causes your immune system tries to fight.
That's right.
And these are called T cells that are these
among many others, but yes,
many other cells that try and go and fight against cancer, and then they have a difficulty targeting the cancerous cells because they don't necessarily, the cancer cells are like camouflaged with the other cells, and so it's difficult for them to find them and kill them. And so then this is a very interesting way to try and leverage the human body to be able to identify those cancerous cells and kill them so we can live a little bit healthier for longer. So what is exactly, how did you discover this process at PrimeVax? How are you really incorporating the T cell?
Absolutely.
This is fascinating.
No, thanks so much, Alan. No, I love the fact that you're actually incredibly well read up on the topic. So kudos to you, man. I love it.
Okay, Red.
I love it. No, I love it because a lot of people, when they talk about cancer, I think they speak in terms of vague conceptual ideas of what it is. But when you actually get down to the cell-specific type interactions, or how the immune system relates to the cancer cells, for example, and how that interaction occurs, the combating the constant cat and mouse game. The fact that you're diving into the detail level, I love it. So, our company, the idea behind it is very, very simple. Reprogram your immune system from the ground up to basically remember or to be what it always was before you actually had cancer to begin with. And you need to basically crash and reboot the entire immune system to do that. That's the overarching idea. So what we do is first we have to do what's called a dendritic cell vaccine. These have been tried in the industry. Many people have actually done this for a decade or two.
What is a dendritic cell?
A dendritic cell is one of your main immune cells in your body right now.
Is it in my blood?
It's in your blood.
It's in my blood.
Yep. It is basically.
Is this smaller than a red blood cell?
It's a white blood cell. It literally is a white blood cell. Yep.
So a dendritic cell is a white blood cell.
Yep. So
and
basically,
there's like, what is it? 5 times less white blood cells than red blood cells, or even less than that?
Oh, that question is above my pay grade, my friend.
But there are less, though, right? Okay.
Yeah. Generally speaking, you don't need that many dendritic cells in the body compared to red blood cells, which might deliver more nutrients or what have you. Your dendritic cells are there to keep you safe. They're there to act as the primary scouter or scout cells of your immune system.
Yes.
Anytime that your body finds something wrong, and then it tries to induce some sort of an immunological response, that's your dendritic cell working. Mm-hmm. So the easiest example I can think of to show people is suppose, I'm going to roll up my sleeve for a moment here, right? Mm-hmm. Suppose if I were to just press and, kind of flick my, wrist a little bit, right?
Yeah.
Scratch it up, get it all bruised up, right?
Yeah.
And, we'll just keep talking for a little bit.
Yeah.
But when we come back to it a couple of seconds later, you'll notice that, my wrist is starting to turn red, right?
Exactly, yeah.
So that's basically like an inflammatory response. And then later on, your dendritic cells will actually go there, find what happened, and then communicate back in your lymph nodes and talk to your T cells and say, you know what, there were bad signals here. Something funny is going on. Go there and kill whatever you think looks bad. And here's what the information looks like that is relevant to the bad, damaged cells.
And there's a certain list of inventory that they have that they know of what constitutes being bad.
Exactly. And they've identified that by physically going there, surveying it, and then going back to the lymph nodes and telling the T cells that. Mm-hmm. That's basically what the process is.
So they have to take a step through the lymph nodes before communicating to the T cells.
Well,
because lymph nodes, they circulate the white blood cells. What do they do with the white blood cells?
Lymph nodes are basically one of your primary immune functions in your entire body. Like you have lymph nodes throughout your entire body right now.
Yes.
It's where a lot of your T cells reside, actually.
Okay.
So you can think of them as like the police depots in your immune system. Meant to go and, like when the dendritic cells go to them, the T cells that are sitting there in dormant status reawaken based on the information that the dendritic cells provide the T cells, and the T cells say, hey, there is something bad going on here. You've given me the information. Now let me go to that location and kill whatever that actual thing is that you gave me.
The police depots is so funny. And then they're the police cars that go out of the depots.
That's literally it. And they have a list, based on what the dendritic cells provide them. The dendritic cell says, oh, this looks like a bruised skin cell. It's in this general location of this arm. So that my T cells are probably right now going via my bloodstream to go to where that location is and trying to find whatever the damaged cells are and kill them. So that's how your normal body functions. And you and I, knock on wood, are pretty young and dumb, and we don't have cancer, thankfully, to our knowledge right now, right? But for a cancer patient, that entire process I described has already failed them. And that's where we come in. That's where we think about, in order for you to...
Which part? The police depot, the communication of the police depot?
All of it.
To the lymph nodes?
All of it. There is some sort of a failure in the entire systemic process.
Okay.
So basically, once the T cells go there and kill the bad cells, right? Then the healthy cells surrounding it are saying, well, okay, there's some leftover space. Let me fill that in with healthy cells again. And then macrophages and other immune cells come in and gobble up the dead cells that the T cells killed that were damaged. And that's what cell cycle rejuvenation is, as one archetypal example. That's how your body stays healthy right now. So for a cancer patient, in order for a cancer lesion or a tumor to exist in the body in that particular location, what that means is that entire process, because it's one big loop, somewhere in that process, something was disrupted. And most of the time, it's probably in a couple of different places that this disruption happened. So I'll give you an example. As you mentioned, cancer cells are really good at evading and hiding from the immune system, right? A cancer cell might pump out a protein called TGF-beta, and the TGF-beta will literally act as like an invisibility cloak. It'll basically hide that cancer cell from the immune system, when the T cell comes there, knowing the actual correct information of what that cancer cell looked like, because your dendritic cell already found it, went back to the T cell said, oh, I'm ready to go, I'm gonna go there and kill it. T cell gets there, the TGF-beta puts the T cell to sleep. And then the T cell can't do the killing. So you have a whole bunch of T cells sitting around, either sleeping, napping on the job, or what have you, right next to the tumor. And in fact, those T cells are what we call in the industry immunosuppressed T cells.
Immunosuppressed, yeah.
Yep. So
wow. So that protein causes the T cell to fall asleep on the job,
basically,
right next to the tumor.
Yep. And
that's so shitty.
That is really bad.
They're right next to it.
Yep.
I mean, it's not like it goes back and
the gun is loaded with the pre-programmed bullet to actually kill that cancer cell. The T cell is ready to pull the trigger, and then all of a sudden, it just goes off, right? And if that's the case, you know, you're in that scenario, right? Your immune system has failed you because the TGF-beta pumped out by the cancer cell has overcome the stimulation, the immunostimulation or immunoactivation status of the T cells going there. So our entire approach is to say, let's disrupt all that and let's make the tumor microenvironment re-stimulated again. But first, we have to begin by making the dendritic cells do what they were supposed to do. So another example in this entire process is that, you know, sometimes your dendritic cells are not able to find the actual cancer information. So remember the latter example we mentioned was TGF-beta, right? The T cell goes there, can't kill the cancer cell. Well, it could fail earlier in the process as well. Yeah. Your dendritic cell goes there, tries to find the cancer cell, doesn't find anything, or that maybe the TGF-beta or any of the other proteins that the cancer pumps out might cause it to evade the dendritic cell itself. Then the dendritic cell never actually picked up the information, or the dendritic cell picked up the information correctly, but then was also switched into immunosuppressed status as well. So when they go back to the police depots in the lymph nodes, right, and then they talk to the T cells, they tell the T cell, well, here's the information, but don't really bother like going there and killing them too hard. They're okay. So the cancer cell basically tricked the dendritic cell in that scenario to not really relay the correct, you know, killing signal information to the T cell. Then the T cell itself learns the immunosuppressed, you know, personality type, or, you know, whatever analogy you want to use of that dendritic cell that was also kind of, you know, like immunosuppressed. And then when the T cell goes back there, it doesn't really have any motivation to actually kill the cancer cell either. So there's multiple failures in the entire, you know, like cancer, like carcinogenesis / you know, cancer growing process that your immune system has failed you on. So we first have to make sure that the dendritic cells from the get-go are programmed correctly. So for us, what we do is we're going to take a sample of the patient's own tumors via a biopsy and a sample of the patient's blood. And from the blood, we'll isolate the white blood cells, which contain the dendritic cells. And we're actually getting basically like what are called immature dendritic cells. You can think of them as just baby dendritic cells. And we do a process in which...
And you can get those all the way through their 70s or 80s.
Exactly.
There's always dendritic cells.
Your body always has dendritic cells. And it can't not have them.
And these nascent dendritic cells, these early stage ones.
Yep. So basically, as part of dendritic cell growth, they begin as what are called monocytes. And then as they grow into you know, adulthood, they turn into one of two things, dendritic cells or macrophages. Dendritic cells are the scout cells that we described. Macrophages, you can think of them as, the easiest analogy is like they're kind of like Pac-Man. They just gobble up whatever damaged, you know, like ghost type cells that are sitting there. If you have some sort of a damage signal, like for example, when I bruised my skin earlier, and the macrophage is what comes here, chews up and, you know, basically eats up the damaged cells and then goes on their merry way. So your white blood cells, those baby monocytes, can be one of those two things. Our process is we heavily skew your monocytes toward the dendritic cell class. And we pump them full of as much immunostimulation as we can while we put them in the presence of the cancer information that we got from the tumor biopsy. Because we're literally surgically removing a piece of the patient's tumor, grinding that up, and then when you grind that up, the tumor cells release their antigen or cell ID information. They must. As part of their dying, the dendritic cells see that, pick it up, and are basically matured in the presence of what we think is probably one of the best immunostimulant cocktails in the industry. And obviously, part of that is our own internal know-how and patents and so on and so forth. But what we end up with is within less than a week, basically little vials of finished dendritic cell product that are matured, that fully recognize the patient's own full personalized cancer information.
That can get past that protein inside
the TGF-beta.
The TGF-beta inside of the tumor that's repelling away.
We think so. There is.
Okay,
I mean, that's what we need to prove in the clinical trial.
Trials.
Exactly.
Cool.
The preclinical data heavily suggests that we think that it should do that. But at the end of the day, where I come from, I think that human data is always the gold standard. Nothing else matters.
I love how you gave the story of how you take the cancer biopsy and then you grind it up.
Yep.
So that way you get the cell ID. That's a really good way to say. And then if you, like you said, if you're skewing all more of the dendritic cells from the baby white blood cell, then you can even boost it up further into a state of immune prosperity.
Absolutely.
And then that makes it even easier to combat potentially. So this sounds very promising. Okay, and then is a good next step into dengue, or should we wait?
Yeah, absolutely.
Okay.
So basically, let's assume that we've done the hypothesis correctly, that we've built a very good immunostimulated dendritic cell product that is able to actually recognize all the cancer that the patient has, right? So now if you think about it, that means that specificity of the patient's own tumors has now been correctly reestablished in the body when we put those back in.
And then is that just through a normal blood injection?
We'll do it via an intravenous infusion.
Intravenous infusion one time or through a couple days? Or
that really depends on the clinical trial design, but it will be either one time or 2 times or 4 times, depending on the specific arm of the hypothesis we are testing.
Okay, cool.
So part of that is going to be understanding the schedule, and understanding the schedule is literally what you do as part of running a clinical trial. But the hypotheses are laid out there so that we can do it over like a week or up to 2 months, for example. So suppose we've done that, right? Well, at that point, you also still need...
I have another quick question.
Please.
So how long would it take until you've administered it intravenously to know if it has worked?
Yeah, so we're going to basically give the patient about a month and a half before we do their PET and their CT scans.
Okay.
And that's when we actually get the first real response data.
Okay.
Now that said...
It could start working on it right away.
Absolutely.
Exactly. But for actually taking a whole bunch of cancer cells, and that might take a couple of weeks.
Yeah.
But it might start working on it right away. Okay.
Yeah. So in fact, it's interesting, Alan. I love the fact that you're actually drilling into this because what you're describing is what we call in the medical industry as you don't want to have what's called a false positive too early.
Oh yeah.
So sometimes one of the main issues that immunotherapy drugs run into is that because their entire premise is that they're working on actually stimulating the immune system to overcome cancer, right, and kill it, sometimes if you take a PET or a CT scan too early, you might be picking up a false positive in which you're seeing the cancer actually growing more. And so because what you're going to be doing before you give the patient the treatment is you're going to do a PET and a CT scan, right? You have to first see how much tumor is in them before and then how much tumor is in them after. Well, if their after picture is measured too early, you might actually read out a whole bunch of T cells and a whole bunch of other immune cells going in and doing their job, fighting the war against the cancer cell in that region, right? In which case that scan might come back and say, actually, no, your tumor grew, but that's a false reading. So you actually want to give the immune system a little bit more time to actually go back to homeostatic control, for example, go back to more of a baseline level, and then do the correct scan.
And then that's interesting, and that's usually about 4 to 6 weeks would give it enough time.
I think so.
And then what would the PET or the CT scan, what does that look like for imaging? What is the type of imaging that's used? Is it electromagnetic? How does it actually come in?
Again, that's a question way beyond my pay grade as well.
Do you know an approximation?
I mean, basically all I know from I'm not a radiologist, I'm a business guy, And but what I do know about the entire imaging process is that basically they just shoot a bunch of lights into you. The lights are specifically looking for certain like masses based on certain markers or what have you, and then those things are going to be the thing that you do to establish the baseline, and then you do the exact same measurement again for a post-treatment.
Yeah, and so then the lights are then trying to find the masses and different masses of different cells, and then that'll tell you what kind of cells are there, and so certain cancerous cells have a different mass. Then.
I think so.
Okay. Yeah. Interesting. Okay. I want to dig into that more.
Yeah, absolutely. That's more of an education for me as well.
Good.
That would be great. Yeah.
We all are slowly learning this stuff. Okay.
Absolutely.
All right. So yeah, where are we now?
Yeah. So now at this point, let's assume I've gotten the specificity down. Well, you have to understand from our perspective, like where we think about cancer patients is that if you are already a late-stage cancer patient, and that's the patient population we're going after, we want to treat patients who've already failed other treatments, for whom those other treatments have failed.
But why not treat people that have the very, very first signs?
We would love to. Yeah. But this is actually part of a regulatory strategy.
You might be more successful with the very first signs.
Everybody tells us that, Alan. You're absolutely right. Oh man, I would love to treat earlier cancer patients, and we will. We'll get there. But from a regulatory strategy perspective,
they only let you test people that have failed everything?
Yeah, and the reason for that, well, I wouldn't say they only let you, but your regulatory road, when you deal with the FDA or any government agency, and I used to work in the Department of Health and Human Services, I can tell you this, you want to play the game according to the rules defined in the industry. And their rules are, they think of as, number one, there's already standard of care out there for, decently late-stage cancer.
Oh, sure.
Those are well understood and well studied with many, many other clinical trials.
Versus your new stuff.
Exactly.
How is it going to compare?
So so what you really need to show is basically for the patients who failed those ones, do you add something else to the process? Yeah. Do you really push the needle forward a little bit? Yeah. And if you do, honestly, from a pure humanity, exactly. They'll let you in, and from a pure humanity perspective, right? If you want to think about this as a humanitarian effort, which I think is really important when you're in cancer, the bottom line is that, any movement that you make for a non-responding patient population means that the entire cancer patient population is benefited.
Definitely.
Right? Yeah. And you want that. You want as much coverage as you can possibly get for as many people as you can. So we'll begin with late stage. We treat those patients, and then we slowly work our way back in time.
Yep.
But to answer your question, if we're going after late-stage cancer patients, like right now in the very, very short term running this clinical trial, well, those patients have been already hammered by many other drugs, right? Yes. Chemotherapy, other immune therapies. Their patient, like their white blood cell counts might just be lower, for example. Their immune statuses are weaker than you or I, right? Yeah. They need something, like a kick in the pants, basically, to reinvigorate their immune system to a much higher activated status level. So if we've defined the specificity using the dendritic cells, we need to have something that greatly stimulates the immune system, and that's where we think dengue virus comes in.
This is crazy.
Yeah, it is.
This is crazy.
Our entire hypothesis is literally the exact opposite of every other biotech out there. Everybody else wants to finesse the problem. And we totally welcome that. We think that they should. But we're saying, let's see if we can shortcut the entire process and brute force the immune system instead. Now, so what we're going to do is we're going to inject cancer patients with the dengue virus. And we have a strain of virus.
Pre-intravenous.
Correct.
Yeah.
Before we've actually
like a couple of days before?
Yeah, like basically 5 days beforehand. So the process is this, before the treatment begins, we'll give the patient, the dendritic cell making process. We take their blood and their dendritic cells out. You give us a week to 2 weeks. We make the product. The patient is ready to come into the clinic or to the hospital. We inject them full of the dengue. They'll be basically incubating on that for about 5 days. Right when they develop fever symptoms, that's when we infuse the dendritic cells in. And the reason for that is because basically when you get sick with a dengue fever, your body's going to go into a very, very high activation status.
But how do you know that dengue puts the body into extreme immune activation status versus when I just have the flu and my body is trying to combat the flu?
Glad you asked. We've analyzed every single pathogen, every single virus, every single bacteria, every single other immunostimulant under the sun, and we've run experiments on our own dengue virus data, and we've compared it to other data in the literature.
I can't believe you did that for less than $1.3 million.
Oh yeah, that's a separate aside. We got to FDA stage spending $1.3 million.
But how did you analyze all of that with all these pathogens?
Oh my gosh. We didn't have to run too many experiments, fortunately. We ran our own dengue experiments, and then we compared it to what was already published in the literature.
For other pathogens.
For other pathogens and other viruses, other bacteria, and so on and so forth.
So what's the percentage increase for dengue for your body's immune system?
I mean, we've analyzed it, for example, 2 examples that we compare against would be, like herpes and vaccinia viruses, which are 2 of the other viruses currently used as oncolytic virus agents for anti-cancer purposes.
Okay.
And what you'll find is that, their cytokine profile, cytokines referring to blood proteins or, like proteins in your body, their cytokine profiles, which act as basically immune signals for your cells, their profiles are very much like a mixed signal, a mixed bag kind of an approach, where some of their cytokines induced by those viruses are immunostimulatory, and some of the other cytokines are immunosuppressive. So you kind of have a push-pull effect when you have those other viruses involved, but not dengue. Dengue makes everything go into crazy haywire activation status. And the trick for us is actually more a matter of safety. And to me, patient safety is the most important thing, right? Our strain of dengue is coming from a time period that really doesn't even, from a bygone era in the 1980s. You can't even find this strain of dengue anymore. It was isolated in Puerto Rico before the more virulent dengue strains overtook it. So the interesting thing is that we actually have data from the CDC and from the World Health Organization supporting the notion that our strain of virus is very, very mild. I shouldn't say mild, but from a pure medical perspective, there are certain terminologies you're supposed to use. But it's more on the lower viremic, meaning infectivity levels, compared to other, hotter strains of dengue from, let's suppose, Vietnam or Thailand.
Okay, so this is it's a kick for the immune system into haywire, but it's a
mild
more
kick.
of a mild kick.
Yep.
Okay, and then what? So then the
data. Let me just finish that
go
thought,
ahead.
if I may.
Go ahead.
I'll never forget this. We ran our preclinical experiments using basically taking the dengue virus that we have and infecting human white blood cells with it. So which is exactly the infection process that you would expect in a human when a mosquito bites you and you get the dengue fever, right? So we did that, and then we measured, like basically control groups, unstimulated immune cells and stimulated due to dengue immune cells, and then we put them on top of various different tumor cell types. We had 2 different melanoma skin cancer cell lines. We used a triple negative breast cancer cell line, which is known to be very, very difficult to kill. And we also had a lung cancer cell line that's also known to be very difficult to kill. And what we found consistently across the 5 or 6 different experiments we ran on these with dengue is that anywhere where the dengue stimulated the T cell or the natural killer cell, which is a different axis of the immune system, by the way, and we'll talk about that later, the marked increase in ability to kill the tumors was something between like 2x to like 20x more than the control groups in the same 4 to 10 hour periods. And I remember this.
Wow, that's a big increase for a short time.
So basically you just put a T cell, a regular T cell, or a natural killer cell on these tumors, and you see how much does it kill. And then you put the dengue stimulated T cells or natural killer cells on these tumors, and then you see how much those kill. And those greatly overshadowed the ones that were unstimulated. So we know that dengue has this immunostimulatory effect, not just the T cells, but also on the natural killer cells. And just by way of context, in the industry, you have what's called the innate immune system and the adaptive immune system. Your body has both. And the innate immune system is much more primitive. Remember that when I was flicking my arm earlier, right? And everything got all red and bruised? That's your innate immune system responding immediately. Your natural killer cells and other types of cells go there pretty much like on a very, very rapid basis and find what's going on and just kill something. They do it indiscriminately. They do it based on, like for example, if your cell has no ID, no antigen presentation, the natural killer cell just kills it directly like that. T cells are adaptive. They're on the adaptive side of the immune system, which is much more smart, much more intelligence-driven, much more where the dendritic cells interact with the T cells, and the T cells pick up the info, and then they go back and kill the cancer cell. They take a little bit longer to work. So the neat thing for us is that dengue does both of the adaptive and the innate arms at the same time, which not a lot of other products do. In fact, I couldn't find any other ones in the literature that hit on both at the same time with a well-established, understood safety profile the way that dengue has. And that's what got us excited. And I'll never forget this point. After we finished that experiment, there was another third arm of that entire trial, or that experiment design, in which we took the dengue virus proteins that came out of the human blood after the human blood was infected, and no T cells, no natural killer cells, no effector cells that are meant to kill, right? No immunostimulation. And we just put the proteins directly on top of tumor cells. And that killed a bunch of the tumor cells directly as well. So we didn't really quite understand what that meant initially, but I'll never forget this. Dr. Bernie Fox, a PhD, one of the foremost experts in cancer immunotherapy, past president of SITC, the Society for Immunotherapy in Cancer, and acting as an ambassador for SITC now. He's on our science advisory board. He looked at that data, and I remember this here, leaning close to me. He put his hand on my arm like this. He looked me dead in the eye, like head on, and he said, "Tony, this direct apoptosis arm, you know what this means?" And I said, it means that the immune system, that this thing kills it kind of like what a T cell does. And he goes, yes, but it means that dengue works like a drug. It doesn't work just like a virus. You have something here. Get this to clinical trials as soon as you can. And when he said that, in the context of all the other great T cell stimulation that we saw and the great NK cell, natural killer cell stimulation that we saw, and this third direct cell death inducing arm of it, we said, oh my gosh, we are onto something that literally no one else is doing. We have to do this.
Yeah, so dengue acts like a drug as well as a virus.
Yes.
So it can be administered in its mildest forms that have been isolated in the past, which good, they did that.
Absolutely.
And now you can replicate that and then use it as a drug. That's so interesting because people typically don't think about that kind of stuff. So hopefully this works out because this seems like a big advancement. I'm glad I'm glad you pulled me over for that because it was really good
intimate moment.
Yeah, it was good to feel that to really feel
so
that moment of the story.
Yeah.
So okay, so now I go through. Hopefully, in the future, you'll take a blood sample and a biopsy sample, and preferably at an early stage cancer's time, and then, and maybe even one day, even pre-cancer, just in general, I can administer dengue as a drug to combat other types of malevolent activities going on within my body, other kinds of pathology. Who knows what it could be used for down the line? But boom, hopefully we can get you through clinical trials and get you out into the world.
Thanks so much. The funny thing that you mentioned about that, Dr. Franco Marincola, who is also a former president of SITC, and one of the foremost experts on genes and cancer immunotherapy. He wrote literally the textbook on cancer. I have the textbook. It is written with him and the current president of SITC. He's actually really interested in dengue, not just as an immunostimulant for cancer purposes, which I think is very, very good as an opening hypothesis for us, but because dengue has this wide immune response along both the innate and the adaptive immune system, he saw that immediately as a means of combating many, many other diseases in the future. And he thinks that, from an immunostimulation perspective, there's so much good things happening, pro-immunostimulation, that you want to just try this out on a whole bunch of other stuff as well.
Exactly.
And to your point, we do want to go much earlier stage in the cancer patient journey. We want to go before they receive standard of care. Ideally, in the very, very long run, the vision for us is to turn this into a prophylactic vaccine. And that's part of us building out the clinical trial data sets and justifying the safety to be able to go earlier and earlier in time,
of course,
and dropping the cost curves down as part of the drug development process.
Of course.
So,
nice.
So the tip of the spear is late stage cancer, and then the spear spreads wider, and we go into all these other different directions once we get some early good data.
Yeah.
Yep. So that's what we're gonna do, man.
Hell yeah, PrimeVax. 3 years in, less than
3 years,
like less than $1.5 million on getting to clinical trials. That's a big deal. That's tough to do. So congratulations.
Thanks so much.
So okay, so that's PrimeVax. Now maybe we ask you some thought-provoking questions about from our show, but also about the industry very specific to you that I think maybe you'll find interesting. What do you think about the ethics of genetic engineering? For example, let's do genetic engineering, intelligence or metabolism augmentations, and how those will be very available to people that have more money up front.
Yeah.
What do you think about that in general as an ethical consideration?
I don't think so my take as a drug industry guy and as someone who came from pharma and biotech, I would say is that, like that's just how the industry works. You have to allow the early adopters with any sort of a new technology, iPhones, laptops, the internet, right? The early adopters are the ones who are most gung-ho, most excited, and who are willing to pay the money to actually do it, right?
Okay, and then the cost curve will drop down.
Exactly.
Okay. But so what do you think about the ethics in general of you being able to genetically engineer whatever you want on yourself, or potentially do some biowarfare? What do you think about this? Should there be controlled experiments or studies on the health effects on one person over time?
Yeah, absolutely. So the FDA already has a very, very well-established process for new drugs going through development, for which, CRISPR, Talons, all these new technologies that are coming out for gene editing will need to abide by. And we know that they will need to abide by those rules because there were companies who were trying to skirt the line. There was one out in San Diego recently, I read on the news maybe half a year ago, in which they were trying to basically justify the notion that, like their drug or their treatment process did not follow FDA requirements for what constitutes a clinical trial. So therefore, they did not need to go through the FDA process. Well, they got slapped down very, very hard with a cease and desist letter.
Some process.
Yeah, exactly. I mean, like the FDA shut them down, made them go through the entire rigmarole. And honestly, they pissed a bunch of people off in the FDA, and you do not want to piss people off in the agency.
You won't succeed with whatever else you want to push through.
Yeah, and for better or worse, I think this is another question that people tend to ask. Like, well, the U.S. FDA is too strict. Like they don't do things that.
What's the right amount of regulating that enables innovation?
Exactly, absolutely. And
what do you think?
Some people will go to Brazil or other countries where the regulations are more lax, right? And they'll get their treatment. I think that for better or worse, the US FDA is the best standard of high quality data and quality, ruling in these types of spaces. Yeah. And I say that honestly because all you have to do is look at every other country that has an established healthcare system, all the European countries, Japan, even China, like Australia, Canada, all these places, right? They all defer to the FDA in the US on drug quality. And they actually generally accept more US data going from the US to those other countries than the other way around. Yeah. Meaning, if I run a clinical trial in Italy, I try to bring that data into the US FDA may not accept it.
You have to run another one maybe here or whatever.
Yeah, exactly. But conversely, the opposite direction is much easier. Exactly.
So then how do we work together across the world, especially what are your thoughts about the way that other countries like China are putting a lot of attention on figuring out how to work on fixing pathology, on living longer, on living healthier. How do we work together but also still foster innovation with this movement for globalization?
Yeah. So speaking of China, I just learned this recently. Apparently, all immuno-oncology clinical trials are halted in China as of like a month or two ago, with the exception of one specific region in like the Hainan district. And the interesting thing is that actually the Chinese government says, you know what, there have been too many drugs going through the clinical trial process here in China. There's 10 PD-1 drugs that were already approved there. I forget if it's PD-1 or CAR-Ts. I want to say it's PD-1. Don't quote me on that. But many, many new drugs have been already approved in China, and the Chinese government said, you know what, this is too much for us to handle. We're just going to do a blank slate, shut everything down except in this special region. And in this special region, as long as you meet the bare minimum requirements, you can run whatever clinical trial you want. This just happened in the last couple of months.
Okay, so what was the reason why again? Because in these other regions,
too much oversight needed from the Chinese FDA.
And then they just said, only in this one region we'll put all of our oversight.
That's right.
And then everybody goes to that one region.
Exactly. And then you have a much more standardized pathway there. And then
so that became their FDA kind
Exactly, exactly. So but that regulation doesn't apply here in the US at all, right? We go through a big process where you pick your clinical trial site throughout the country, you get that written up into a protocol, and then you submit that to the FDA, right? That's the process. But in China, they're just saying, yep, the special region, and that's it. So the interesting thing I would say to answer your question, how do we work together with them? We, as biotech companies, if you were to actually take the industry view on it, which is, like most of the pharma companies, the Mercks, the Amgens, the Genentechs of the world, and you ask them, like, do you want to get into China? They'll say, actually, no, like, I don't want to. It's kind of a dumpy market, It's hard to get into. You have to know all these different special connections and everything else in order to get anything done in China. But the interesting thing is that, with this new special program, I think the onus is on us as the industry to actually be more open-minded about this type of stuff.
Definitely.
Because
across the world.
Absolutely.
Yeah. To work together with all the brilliant people.
Oh, for sure, man.
Yeah.
I mean,
because all of a sudden, if other countries in the world know about dengue,
yep.
Then maybe they can be like, well, what other things exist like that in the world that can be used as a drug?
We want everybody else to be doing that entire process. We want to foster that level of imitation. Mm-hmm. Because if people are imitating you, you're doing something right.
Yeah.
That's how I think about it, right? So absolutely, we want that. We want to share the data. We want to be, like a very, very transparent organization to everybody else, like the FDA, hospitals, doctors, but most importantly, the public. And the truth is, I think from my perspective, I don't think that the pharma and the biotech industry have done a very good job of actually being consumer friendly. Mm-hmm. Like, I think a lot of people out there in the world view the pharma and the biotech industry as this big, giant, monolithic, like black box for which, they're hiding, for example, the cure for cancer tucked away somewhere in their back pockets or in their laboratory somewhere, and they're just building drugs that are meant to sustain your life a little bit more and more and mm-hmm. more, right? And
we'll never actually know.
Yeah, I mean, I don't think we will, but...
Because if you have $1 trillion, you can basically do whatever you want with your health. You can probably even live forever at this point.
You would think so.
Yeah.
I think my personal view on that is that, there are going to be certain limitations to human biology. And there are also going to be certain limitations to how much we can actually control everything.
Definitely.
But that's, part of the... That's part of the...
Soon that won't be the case.
Oh, absolutely.
Yeah.
But that's part of the game that the smart, finesse biotech people are doing, right? We want them to be going after CRISPR, to build all those things out, to do all the gene editing, be very, very laser focused on editing your T cells, for example, and then just curing disease from the ground up. We want that for sure.
Let's end on a couple questions.
Yeah.
How about, Tony, do you think that we're alone in the cosmos?
Oh man, I really, really hope that we're not.
Why, you want some friends out there?
I think we do. I mean, the truth, it would be a very, very lonely existence if we were the only planet circling the only sun that happens to harbor, life, right? And I want that contact. You look at Star Wars, Star Trek, Starship Troopers, pick any of those science fiction things with the star phrase in front of it.
Why do you want the contact?
Because I think it would be interesting. I think it would be, honestly, it would be something more than what we have now.
Exactly, and it would. I feel like it would instill finally a sense of tribalism across the planet. Finally.
Oh, absolutely, man.
Do you think that we're in a computer simulation?
I thought about this long and hard. I mean, your show is called the Simulation Series, right? So the political answer is absolutely yes, we're in a simulation. But now my own personal feeling on it is that, honestly speaking, if we're in a simulation, we're in a good enough one that the rules are actually pretty well understood.
Absolutely.
And if if I come at you and I punch you in the face, your face is going to get bruised up, right? Yeah. That's just those are the rules of the universe,
and then
right?
There's another rule, which is then you go to jail. It's like
exactly.
It's so funny.
Yeah, exactly. So you know exactly what the sequence of actions or events are going to be when you do these things, right?
You know your next algorithm is to eat food and drink water and use the bathroom.
Absolutely.
Your heart beats and beats and beats.
We process oxygen, we take in oxygen, and we pump out carbon dioxide. Plants are the exact opposite, right? You break those rules, you die. So I don't really care if we're in a simulation or not. I think we're in one, but it doesn't really change the fact that we have a lot of stuff that we need to do to change the world.
To thrive and prosper. But
Absolutely.
The rules are very fascinating. The programming language was very fascinating. And what's really interesting is that the programming language changes based on the mass of the planet, the proximity to the star, the carbon-based life forms or other DNA-coded life forms that have prospered on that planet.
The speed at which you're traveling, all that stuff.
Yeah,
absolutely.
Exactly.
It also changes when you figure the game out. They fuck it all up and change it. I just want to throw that in there, simulation, guys.
Every single time that you learn more about the programming
changes.
That's right.
The programming changes a bit, yeah.
That's top secret information right there. I'm going to get in trouble just for saying that.
Oh, man, don't let the NSA know.
Okay, how about, Tony, all right, how about last question. How about, what do you think is the most beautiful thing in the world?
Oh, man, the most beautiful thing in the world. Probably the notion of earned happiness. And I'm a big proponent of that. I think that I think first of all, beauty is obviously, defined based on whoever is the observer, right? But I think there are some universal constants that you can generally agree on as people. One example being that, people generally have this innate drive to do something, to achieve, self-actualization, right? According to, the Maslow's pyramid, the hierarchy of needs and all that stuff, right? And I think at the very, very top, self-actualization really comes down to earned happiness. And when I say earned happiness, what I mean by that is you have a very clear goal in mind. You know exactly why you're accomplishing that goal. You do everything in your power to make that goal happen, and then you see it and you bring it into reality. That, for a lot of people, doesn't happen often. Some people never get that feeling in the world throughout their entire existence. And honestly, that's what I'm chasing after, man. To me, that's the most beautiful thing.
Yeah.
You do that. Yeah. That is a natural high. You will always feel that. You will always know that you did that, you earned it. No one can ever take that away from you. And I think that's the most beautiful thing in the world.
That's beautiful. Yeah. Really well said. That self-actualization, this earned happiness, this journey, this hustle through this journey of what maximizes my thriving, my feeling most alive, and then how, if that also has some sort of corollary with bringing value to the world at the same time, oh man, that's just there's no regrets then.
Absolutely.
That sounds amazing.
Live a life with no regrets, totally no fear, totally just absolutely this is the right thing, makes sense, no internal inconsistencies, that's what I'm all about.
Yeah, and hustling in that direction. Is that no internal inconsistencies?
I love that.
That's really good. That's well said. I love it. Tony Chen, thank you so much. PrimeVax.
Thank you so much, Alan. It's a pleasure.
Yeah. Awesome job. Awesome job with the company. Thanks for explaining this so relatably on the show, bringing us down to a way of understanding our bodies and understanding cancer, understanding immunotherapy, understanding all of the possible futures, all these processes that exist in getting there. So thank you very much. And this has been a pleasure. Let's get these conversations out to more people. Let's get them more inspired to have them.
Change the world. Change
the world, baby. Thank you everyone for tuning in. We greatly appreciate you. Please share this content with other people around you. Let's get more people inspired talking about this. Like, comment. We'd love to hear from you. Subscribe if you haven't yet. Hit that subscribe button. Gently press it. We'd love for you to join us.
Or smash it, whatever you
Or
prefer.
smash it. We say that sometimes as well. Thank you for supporting us on Patreon. We need to keep the studio thriving, keep the lights on, electricity going, all that stuff. So please do join us on Patreon. That helps a lot. Thanks to our partner producer, Ron Vargas, our partner photographer, Lisa Sinelli. Thank you so much, everybody, and we'll catch you again soon. Peace.